Good morning to all of you, and I'm Anja Haverić. I also come from the University of Sarajevo, but from the Institute for Genetic Engineering and Biotechnology. And today I'm going to talk about air pollution in Sarajevo as well, but using plant models. So basically, this is the research that we have conducted. It was supported by the Environmental Fund of the Federation of Bosnia and Herzegovina, and significantly contributed by Bertrand, both theoretically and practically, as you can see. So you heard a lot about the numbers and the air quality and monitoring of the air quality. So I just wanted to show you this image for two reasons. One is just to take the view of the city, and the other one is probably nowadays it won't be easy to take this scene. So luckily, since the movie Walter Defends Sarajevo is among the most watched movies ever, so luckily the scene has been seen by the numbers. So having in mind the overall context of air pollution in Sarajevo with the high death rate caused by the air pollution, and since we are among the poorest urban areas in Europe in regards to air quality, we have started the series of biomonitoring experiments, and one of them included plants. So why to use plants as biomonitoring models, and what are the advantages of using plants? Well, definitely, as for lichens and mosses, plants are sessile organisms and continuously exposed to changing environment and conditions. Additionally, you don't have to care about lifestyle factors. And of course, the ethical aspects are much more relaxed when it comes to plants. Additionally, due to the high surface of the leaves and the roughness of the leaves, higher plants act as some kind of biological filters in the environment. So that makes them the perfect models to monitor air pollution. So what was the aim of our study? We wanted to find out the new model for plant Comet Assay, and we wanted to identify a plant that is available through the whole year, that is also widely distributed, that has suitable genome size because it affects the plant Comet Assay, and it also can live in different soil types and different environmental conditions. So we wanted to check if Ligustrum vulgare or wild privet or European privet is suitable plant for a plant Comet Assay. So we want to evaluate its reliability, and if it's reliable, to test seasonal sampling period, leaf position, and locational differences in DNA damage. Since the COVID-19 outbreak happened during the project, we could also monitor the effects of the COVID-19 lockdown on the air quality and consequently on the DNA damage in our plant models. So we decided to collect, we first started with the campus site to establish the model, to optimize the methodology and so on. So that's why the campus area has the most sampling samples taken from that area. We also included one additional urban site, which is Pafalići area close to the busy road. And as a control, we collected the samples from Barica region, which is a recreational area close to Sarajevo. We were collecting both adult and young leaves, outer and inner layers of leaves. The periods of samplings were through the 2020 to 2022 in May, June, and December. And since we were doing this in some series and successions for some periods or some localities, we do not have all the samples taken, but you will see that later on the graphs. I know that Amaya is having a presentation on Comet Assay, and I won't steal the show, but I just want to give you a short overview just so you can follow me. So the procedure for the Comet assay, which is a simple and versatile method for the investigation of DNA breaks. You have to collect cells, then you then you isolate them either by detaching, isolation, purification, embedded them in low melting point agarose and spread them over the normal melting point agarose covered slides. Then you do the lysis, DNA unwinding, electrophoresis, and further on you stain them and visualize them. It's basically the same in a plant Comet assay, but there are some specificities in the Comet assay. That means that lysis, step of the lysis is not needed, and you can all find it in this paper and the protocol. So the specificities are that you have to do everything in dark and cold environment. You have to be quick with mechanical isolation of nuclei by short chopping with sharp razor blade. So that's why the no lysis is needed. And you have to do everything in a dark room safe environment. So the method itself is called by the specific appearance of the nuclei, especially if DNA and that is damaged. And the common descriptors used in Comet assay are tail intensity, tail length, and tail moment. And in our study, we measured tail intensity. So, results. We didn't measure the air quality. So since chemists do that and we're biologists, but we were lucky to be able to take the air quality data from the US Embassy website. So they collect PM 2.5 concentration from the continuous monitor located in the US Embassy close to the two urban sites that we were taking samples from. And as you can see on this graph, the highest concentration of PM 2.5 is in December of 2 years. In 2020 and 2021, in March, we had a partial lockdown. And after that, with the release of the COVID measurements, we had a drop in PM 2.5 concentration, but this is also related with the end of the heating season. So on this slide, you can see that when we compared 3 localities in 2 years, we had significant differences for the rural side, so meaning that the Barice had significantly lower DNA damage in leaves, especially in 2022, it was especially observed for the adult and otter leaves. So here is the comparison for the campus area, where we said to have the most collection periods and collected the most of the samples. And as you can see, we had several categories of leaves: inner adult, outer adult, inner young, and outer young leaves. And for the whole monitoring, we can say that adult leaves and inner and outer leaves have significantly higher DNA damage than inner leaves and young leaves, which was obviously expected. So to summarize, Buffalo site, which is heavy traffic area, had the highest DNA damage, and we confirmed that as we accepted. And generally, outer and adult leaves showed higher values of tail intensity. So since we observed the lower tail intensity at Barice site, we assumed that Barice has better air quality, but can we be sure about that? Since we didn't measure the air quality at this site, it's still questionable, and there is no monitoring station, air quality monitoring station close to this site. So I would probably ask Sabina and colleagues from Zagreb to maybe do the measurement in this site. And also we sporadically observed the increased tail intensity in younger leaves, and we assumed that it could be related to the differentiation of leaves and then dynamics in leaf development. So another thing to investigate and to check out on that. Additionally, when we did the correlational studies, linear regression analysis between tail intensity values and average PM2.5 concentrations, we found positive association, but not significant. So we also assume that we have to include other pollutants and other air quality data to test for the correlations. So to conclude, our plant model was effective, and it can be used to assess the impact of air pollution on DNA, on plant DNA using plant Comet assay. DNA damage in leaves of Ligustrum vulgare depends on the sampling period, leaves position, and stage of growth. And DNA damage can be also correlated to the three successive years, is aligned with the COVID-19 lockdown periods and general improvement of the air quality. So limitations of our studies were related to the missing of additional air quality data and missing of detailed meteorological data and environmental conditions.